Good morning, everyone. First of all, I want to thank the Kariman Project for inviting me to give this talk on stranded cetaceans as a unique opportunity to study populations from a biological and medical perspectives. My name is Gabriela Hernandez, and I have been in charge of the detention and post-mortem investigation of stranded cetaceans in Costa Rica since 2004. Here is some information of my background. The Kariman Project aims to strengthen the network of stakeholders involved in marine mammals conservation in the wider Caribbean region. One of the objectives is to strengthen capacities among the Kariman members, and this presentation is the fourth of four dedicated to marine mammal stranding network strengthening. Stranding networks brings together scientists and volunteers and coordinate their response when an animal is found stranded on the coast or distressed at the sea. You will find here some guidance and data and sample collection on stranded marine mammals. The agenda for today includes a brief introduction, data and samples to collect, field materials and general tips, how to make this data collection and how to make the sample collection, as well as how to storage them. And it also includes experiences on stranding attention in Costa Rica. I cannot start this talk without mentioning Dr. Joseph Darassi, who is the pioneer in the stranding of marine mammals in the world and who passed away in 2015. His heritage includes, among many documents, this book that it has 2 editions. It's like a Bible of marine mammal attention and post-mortem investigation. You must need this one in your personal library. Recently, there are another really good documents for cetacean investigation that are available online, such as this one on the left from ACOBAN and ASCOBAN, including best practice on cetacean postmortem investigation and tissue sampling. On the right, there is a guidelines for coordinated cetacean stranding response during mortality events caused by infection agents and harmful algal blooms. There is another 2 examples of really good documentation on the left, face-to-face with a beach whale, guidelines for the welfare of live stranded cetacean, that is really important that you can have, from the Irish Whale and Dolphin Group, and on the right, marine mammal necropsy, an introductory guide for stranding responders and field biologists from Woods Oceanographic Institution. And of course, the Bible of Veterinary Medicine in Marine Mammals, this book, the CRC of Marine Mammal Medicine. And on the right, there's another Bible for identification of marine mammals of the world. Okay, let's start. The first step in the attention of this animal is to understand and keep in mind that most of the time, the reason of the stranding, even with the investigation, remains as not determined. It is not always possible to establish the stranding cause. Each cetacean has invaluable information for its population, and you need to keep in mind you only have one chance to collect good samples. That's why it's important that information that we are going to see today. There is information that you can collect. You need to keep in mind also that this information can be as complex as you wish. You can choose one of these areas, or you can collect information from all of them, including biological information, health status of the individual, ecotoxicology, or try to describe the stranding cause. Another concept is the normal flora of these animals may be potentially pathogenic for humans. Examples of diseases or zoonoses include, for example, viruses such as influenza that can be transmitted from these animals to humans and vice versa, and bacteria like Brucella septi that can be present in these animals. Also, antibiotic-resistant bacteria from the environment also present in these animals, or parasites like Toxoplasma gondii, or fungi like Paracoccidioidomycosis septi. That is a nasty fungi reported in dolphins and humans. Consistently conducting necropsies permits that countries or regions has a baseline of information. There is some concept that need to be always present, like marine mammals are sentinel of the health of the oceans. Gross observations can guide potential diagnosis in histopathology and other studies as cause of death. These animals can evidence chronic diseases or fishery interaction or emerging zoonosis. That helps to describe trends in population health that can be monitored. This is also important. Get in contact with government staff. Help is always necessary, but if you are going to take blood samples or performing other medical procedures, must comply with state and regional and federal veterinary regulations. Also, you must consider before to start analyzing the animal, do we have collaboration with national or international laboratories or universities? What kind of laboratory has my government? This could be a key point to describe some diseases that are maybe available in your country. How many people can help me to study this case? This is really important from logistic point of view. The second chapter of this presentation includes the information and samples to collect. You need to realize that you're going to make a study of the dead animal like CSI. So you must include the scene information and then the samples to take. The quality of the information will depend on the size, for example, of the animal, the skills of your group, organization you have, and of course, the interest of the team. If you have standardized protocols, if there is available of equipment and supplies, number of animals to be examined, this is important if you have a mass stranding, the amount of time available due to the tide or sunlight, good labeling of the sample, this is really important, we're going to talk about later, and care and shipping and storing samples. There is different levels of information to collect, from information on site, the responder, the species, and some supplementary information include the behavior of the animal prior to the stranding sample that you collect. Here's some example and some listing. And here is an example of data sheet that we use in Costa Rica that we are going to describe today. And this includes first, the site information and the condition of the beach. For example, and everything starts with a responding source, and you have to certify the animal is there. And if the tide, for example, not take the animal away, because you're going to invest some really good effort and money and everything. So you have to be careful and be really have the confirmation the animal is still there. Everything starts with the species, with a photo. This is really important. What is the size of the animal? Is there logistic that support us in the field? The number of animals, of course, and the location. This is important. Also, the tide conditions. If there was some offshore human activity or predator activity that can be related to the stranding, and the weather. This is really important because we are going to be there, and the weather is really important. The time of the discovery, how many hours from the first sight, and the date. Pictures are really, really useful to evaluate the condition to collect the animal. For example, this was a real case on this year in Costa Rica, in Soleil Beach. And they called me and say, yes, there is an animal, it's there. And so the girl sent me this picture, and here you barely see. And so they said, the tip of the dorsal fin is there from a pseudorca. So you need to check the access of the animal with vehicles or even just human access to pull the animal out. If the animal is in a estuary or a river, what are the beach conditions? And if there is predators, this is really important before to get in and take the animal out. The information of the animal, we're going to talk about that later in more detail, but if the animal is still alive is really important, of course. What was the behavior pre-stranding? The animal have directional swimming, it was weak. The stranding, how was it? It was passive, thrashing, or there was an effort to strand of the animal. And after return to the sea, the swimming, how was it? It was disoriented, it was arcing, the animal was arcing or have tremors, that's like a movement, involuntary movement of the animal. There was seizures. So we are going to have two examples here. The first was a calf of a spinner dolphin who receive help, but the animal keep stranding. You can see here the animal is still moving outside the water. Or this one, where the animal was really weak, is still alive, but the animal wasn't in a good shape at all. This is a concept that is really important. You have to keep in mind. These animals, at the beginning, when they die, they sink initially. They can float for days or even weeks. So some of them will strand and outwardly will look unchanged, but internally are really decomposed. Fresh animals can look deteriorate because of scavengers, but still there is useful samples inside. There is carcass classification, and we're going to talk about that later, and there is different codes, numbers from America and some from Europe. But in general, it describes the grade of decomposition of the animal that we are going to talk later. The sheet of information also includes if there was scars present or injury present, is important, where, there is a description space for that, in what part of the animal. If there was some missing parts, for example. For this, you need to take photographs and use at scale. Like this one that we make is a ruler that we make and we plastify so we can wash it. But if you don't have this kind of rulers, you can use a scale reference like your shoe that you can measure later and you can make the estimation of the size later, or like a bottle. This is another example. This ruler that we make, it let us to have information. So you can write the consecutive number of the stranding, the name, if you put a name or ID of the case, and the species. And this is really important that you keep in the photograph so you can follow after years or several animals which photos are from whom. Photographs are really important. Now everybody has a cell phone with camera. So there is a minimum picture that you can get from the animal, include dorsal view, lateral view, ventral view, the head from both sides, teeth, if there was a baleen, pectorals, flippers, and tail. And there's some scars too of other abnormalities. There is some samples that you can start to describe if you take teeth, or if you take baleen plates, how many you got and who has it. This is important too. And this is for future analysis of this kind of sample, basic samples. There is some also in this sheet of information, special measurements or special information for some species. For example, baleen whales, you can count the folds in the lower jaw, so that helps to describe the species. Also, there is really important information on the baleen, so you can write down how many they have, the color, the size. This is important too for the species description. And beak whales are really important. For example, how many grooves they have in the lower jaw, the shape, the size of the teeth, that is important too. So this next sheet has really detailed information and have the metrics that we collect in Costa Rica. That includes 50 different metrics from the same animal. So of course, the species is the first description of the animal, and the second is sex. How you know to describe the sex? Well, females have 3 lines, 3 different slits. Here in the longer one, you have the genital one, and in the lower part, you have the anus slit. But females have 2 different slits for the mammary glands. Males have different presentation. They have 2 slits, the first one for the genitalia and the second one for the anus. These 2 are separated by perineum, and this is important to recognize them. So this is actually how it looks. And you have to realize, like humans, that some males and from some species, for example, big whales or for example, blue whale, that happens to us a couple of years ago, they have mammary slits. And if you are not really careful, you can get surprises sometimes. But this is really the useful way to describe between sexes. Weight. This is really important, but it's not always possible to get it. Of course, you have metrics to do, but there is some, actually the only one, application that you can use. The name is Whale Scale. It was developed for iPhone and iPad by Dr. Craig Harms. And so you put here the species and the length of the animal, and it gives you the approximately weight of the animal. This is important from the logistics and the tension of the animal. Metrics, of course, you need a flexible one and a hard one, so you can use them. You can make some circumference at different levels of the body, and the pectorals at the medium of the body and the tail. The measurements are again as complex as you wish. Like I said before, we collect 50 different measures. This is another example from this book, Marine Mammal Ashore. We collect about 20 different measures. So you make your own decision and how many you want to get. No matter how many you get, you can do it, like you need to do it the same ones all the time. So you can then, after years of information, you can compare and you can get more information. There is another information in this, for example, the blubber size. This is important to estimate the body condition of the animal. So you need to make it the same place all the time for the different species. So basically you do it dorsal, lateral, and ventral, and you measure and you put it down, you write it down. This is important for the body condition. Teeth is important, so you have to describe in more detail, like how many teeth you have in the right side, in the left side, and the upper jaw and the lower jaw. So this is important for description. The third chapter of this presentation includes field materials and general tips. And here is where really detailed information you collect from the animal. You collect tissues for toxicology, samples for microbiology, tissues for gross and histopathology, and for parasites. General tips includes develop a routine for every single animal, do the same. Standardize your protocols. Document everything, use tags and scales. Make a list, a checklist, with the documentation that you need, the sampling material, the safety personal protection material, and for cleanup later when you finish the necropsy. Also, another checklist of field materials you need in your team. And here in colors are represented like all the groups that you need. For example, a live animal, a live and dead animal, necropsy material, and first aid materials. Writing is really important. So use support for writing, and this is really simple, but it's a key point in field necropsies. Choose one person with pretty writing to tag everything. Use bags, ideally sterile ones, like the one on the left, which some of them has a space for writing. So you need here the date, the ID of the case, and the organ or tissue you are collecting. Optional, if you don't have these ones, use like this one, like Ziploc. These ones keep the liquid, like blood, inside, preventing watering. Use nice, fine, or ultra fine point permanent markers. This is really important. Use big and small knife, disposable and not disposable scalpels. And most important, if you use knives, choose one person who all the time during the necropsy time sharp your knives, because the edge, it will lost with the sand or animal skin. One important key point here is if you are working on the field, it's really important that the knife is in your hand or in a bucket for the person who is sharpening. Be careful and do not let the knives in other parts because it can cause accident to your personnel. There is different syringes, size of syringes. The information includes the amount of milliliters that the syringes can collect, the width of the needle, and the length of the needle. Of course, the one that you choose will depend on the thickness of your sample and the amount that you want to collect. For example, if there is fluids in the thorax or in the abdominal area, you may want to know how much liquid is there. For that, there is some laboratory recipients that you can use, but also you can choose plastic bottles and estimate how much liquid is there. You can also use kitchen jars, but of course, after the use, you must throw it away or let them for the next necropsies. Plastic tubes or different capacities are available in the market. The presentation are really important to collect samples. For example, if you want to look for bacteria or viruses, ideally you need the sterile ones. And if you want to make PCR or of the host or parasites or different microorganisms, you need like this tube, which are free of DNA or RNA acids that will affect this will affect your sample if it's present. The histology is the study of the tissue and their structure. For these studies, you need buffered formalin to fixate tissues, but it has a cost on your sample. It will kill your microorganisms and degrade the DNA. So histology need formalin, but the rest of the study and the samples don't put formalin on them. Important to remember, size of the sample matters, and the samples need to be small, no matter the size of the animal. We don't want kilos of whale or dolphins in our laboratories, believe me. Size, if you have a bucket, you put 1 part of the tissue by 9 parts of formalin. This is the formula to use formalin. Buffer it at 10%. So you use these kind of buckets of 3 to 5 liters. In the market, you can find different size of containers, but these ones are ideal that has a lid that is sealable, have wide mouth, and of course the small ones with an extra lid to protect against evaporation. Once it's in formalin, your tissues, you can storage this forever at room temperature. You may change the formalin in one point, but if it's really close, it can be the same formalin several years. Alcohol is really important for disinfection and some samples. You may think 95% degrees alcohol is better than 70, but it's not. The reality is that 95% make a fast dehydration, and some spores, for example, will still alive, so you don't kill them. So if you want a good disinfection, you need 70% alcohol, which makes a slower and better disinfection, and is less flammable and it's cheaper, which is good. So if you have 90% alcohol and you wanted to convert it to 70%, you add 2 parts of alcohol by 1 part of tap water. So like plastic bottles, you can have 2 bottles of alcohol by 1 bottle of 500 milliliters of tap water. Saline solution is another solution that you may need during necropsies. For example, to put parasites and watch them under the microscope. You can buy it, but also there is some tablets that you can add it to 500 milliliters commercial water. So 1 tablet, but this amount of water, and you have it there in the field. Bags, biohazard disposable bags. This is really important. At least you need 10 each, large, medium, small bags per animal. Of course, depending on the size of the animal. It will have different sizes and different kilos that can be there. And this is important because if you have personal, you don't want to full them because it will be too heavy and probably you have to move them to one part to another. So try not be putting here more than 10-15 kilos. Gloves, this is really important. You want that your personal works comfortable and safe. So you better have all the sizes, extra small, small, medium, or large. Nitrile is better than latex. It is more resistant. So in summary, you will need more latex gloves during the necropsy than nitrile ones. So this is a cheaper option. Make your own first aid kit as complete as possible and bring it always with you. Accidents are always possible and you need to protect your team. There is different clothing you can wear, like this one using the US or Taiwan on the right. But no matter which one you have, you need to avoid fluids from the dead animal getting in contact with your skin. This is the ideal one. This is Tyvek material. This is prevent the passage of hazard materials. There is different in the market of different brands, but you have to be aware of the size of the pores so the microorganisms not cross by and touch your skin. The fourth and fifth chapter include data collection and sample collection and storage. Before you start opening the animal, be sure you have help to bury it. You don't want a decomposed animal in front of your house, so you better clean up really well everything. Don't let trash, don't let gloves, don't let bags, and even worse, do not let needles on the beach. This is an example on the right of the organ list that you want to have before start a necropsy. This list, you can use it prior to the necropsy to write in every single bag the name of the tissue that you want to collect. So you don't forget which samples are missing. On the left, this is the list that we use in Costa Rica that is based on the first one, but have more samples. For example, every single part of the brain is separate and stored in different bags. Pathological investigation should take preference over maintaining skeletal integrity for museum collections, and histology samples should be taken of any bone abnormalities that may have present in an animal. The amount of information, it will depend on the decomposition of the carcass. Code 1 are alive animals, so you can collect information of morphometrics, limited life history, external gross pathology, histopathology, diseases, blood studies, and DNA analysis. Code 2 animals are freshly dead, which are organs are in good condition. You can make analysis of morphometrics, life history, gross pathology, histopathology, diseases, toxicology, and DNA analysis. You can make blood studies if the animal has less than 18 hours of death. This is how you take blood samples from an alive animal. And this is how you make it from a dead one. You have to make it intracardiac, so you have enough blood there. There is different tubes, but the most basic ones are with the red cap and purple cap. For a storage serum, you need the ones with red cap. And this is an example of the diagnosis that you can make on field of brucellosis with all the serum that you collect here with the red cap tube. You can store the serum of the animal for decades at -20 is okay. It contains the information of all the microorganisms to which the animal has been exposed in its lives. So maybe you don't have money to run some samples today, but maybe in 1 or 5 years, you can run these samples for diseases. Code 3 is a decomposed animal, but organs are basically intact. You can make for morphometrics, DNA analysis, limited life history, gross pathology. You can make analysis partial for histopathology, partial toxicology. You cannot make blood studies or diseases studies. Code 4, it's an animal that have advanced decomposition and organs are not recognizable anymore. You can make morphometrics, but you can have limited life history. You collect teeth, baleen, stomach contents, reproductive condition through the bones. Gross pathology, barely you can do it. Limited DNA analysis, you cannot make it diseases or toxicology anymore. This is a color of a characteristic code 4 animal. And code 5 is a mummified or skeletal remains only. You can make morphometrics, you can make limited life history, and you can make limited DNA analysis. If possible, put the animal on ice, or you can put it in a commercial freezer. The cost is less than $1,000, it's not much. And -20 to -28 is okay. So this is normally how these freezers works. It has some advantages, like stop decomposition. So you have better quality of samples. And this is really important. Necropsy can be programmed. Disadvantages, of course, the size of the freezer, but the big ones, you put animals less than 2.2 meters. You will have some crystals of ice in the histopathology, which pathologists usually don't like, but it's okay. They can make the description of the pathologies there. And of course, you need defrost time before start a necropsy. -80 freezers keep your samples indefinitely. Of course, it's more expensive. It will cost you like $15,000. And depends on the amount of the animals you storage. We have a rate in Costa Rica of one of these freezers per 10 years of materials. Of course, if you need or you have one of these, you need a really good backup. It will cost you like $3,000, one of 3,000 UPS, but it will give you protection or backup for 18 hours. So you better run and do whatever you need to do to save these samples after that. The correct procedure to start the sample collection, it starts with a big window in the animal. So you better see the whole thoracic part, abdominal part, and you can see the organs on site. There's different procedures, but for example, this is a correct procedure to collect blubber samples for toxicology. So you remove the skin and you let only the blubber. This is a diagram that shows the correct position of the organs. I let it here so you can follow it. Always duplicate samples. We already know why is this. So you have 2 containers for formalin, one for the brain because it will be your biggest sample, and the other container is for the rest of the samples. In the field, you have a cooler and ice packs before you get into your place, into your laboratory. So you put the samples, the fresh tissue samples, in the freezer. So you have 2 sets of samples, as I said before, and the correct time, if you are not able to process it that same day, you can put it in the fridge for 2 days or freezer for years if you cannot run it in the immediately. And the set of the tissue that are for histology or immunohistochemistry, you have it at room temperature for years. So how big should be the samples? We talk about the size. The samples matters. So in formalin, pieces of 3 by 3 centimeters is okay. But the thickness is really important in formalin samples. It needed to be not wider than your pinky finger or 1 centimeter wide. And in the case of the half of the brain, which is, of course, the biggest sample you're going to have, you want to cut them along the tissue so the formalin get in and you don't lose your sample because it becomes rotten. In fresh samples, you can put them pieces 5 by 5 centimeters, and you can put the complete half of the brain frozen. There is transport swabs for bacteria isolation. So you ask your laboratory if they work with this kind of transport media for bacterial protection. There is also some solutions and transport liquid medium to protect viruses, like RNA later, which make an RNA stabilization, or if you doubt, don't add nothing, just frozen or refrigerant. If you want to make PCR of the DNA of the host or look for DNA or RNA of microorganisms, do not add nothing. You can keep the sample in the fridge for 2 days, but again, if you are not going to run the sample under the analysis, you better freeze them. I left here some list of common parasites, the most important ones, but these animals normally has parasites. So you better be careful telling the people the animal died because of the presence of the parasites. Usually it does not. Again, for parasitology, you put them in alcohol if you want to make identification, but also let some parasites without nothing and freeze them. Here's a summary of the different samples to collect and to maintain them from the Marine Mammal Assure book. It's a summary. But there is now a really cool chart that includes a diagnosis that you want to make. the recommended samples, the maximum decomposition code that you can work, the storage recommendation, some solution that you can use, the quantity of the samples that you can that you need, and some special comments. And in the other hand is the organs that you can collect for what diagnosis investigation you want to make. Again, the decomposition, maximum decomposition that you can work, the storage conditions, the quantity that you need, and some special comments. It's a really nice chart. And here is some example of basic necropsy report, which includes by tract the tissue that you want to review. But again, you can make as large as possible, so the information is really detailed. And the last chapter is of the experiences from stranded attention in Costa Rica. Costa Rica is a small country with 1,000 kilometers of Pacific coast and 200 kilometers on the Caribbean coast. The investigation of stranded cetaceans is part of the Wildlife Health Surveillance Program of the National Service of Animal Health. No formal stranding network has been established in the country, and the notification of the strandings event relate in the public and on the efficient communication of the alert to the veterinary professionals in charge. Logistics plays a key role in the successful management of the stranded events of live or diseased cetaceans. The small size of the country and the presence of highly specialized professionals have nourished close collaborations between the general public, governmental agencies, universities, national and international nonprofit organizations, coast guards, firefighters, and private companies. Here are some examples of our collaborators. The interdisciplinary approach between marine biologists and veterinarians has been crucial in the success of the current surveillance model applied. Several techniques have been incorporated to improve the acquisition of biological data and to increase the sensitivity and specificity of disease detection, including biometrics, complete necropsy, serology, classical and molecular bacteriology, virology, histopathology, and more recently, CT scan. And here is the progress that I have told you before. In the beginning, we want to save them all, and we do not protect ourselves. And now, after a few years, we understand what they bring, and mainly is marine brucellosis in the brain. We take samples and extra samples of everything. We protect ourselves. You can see there is no skin part in front of the animal. And so this process results in a success example of interdisciplinary work and One Health approach, and maximizing the scientific information obtained from every case. The research in Costa Rica started with a good sample of serum, 1 drop of serum, and 1 drop of rose bengal. And we got this beautiful reaction. So we knew Brucella was in the animal, but where? After a couple of years, in 2006, we discovered these bacteria in these animals were present on the brain. And that's why they become really weak and has neurological symptoms and die really quickly once it strands. The information opened a wide collaboration between different groups of investigation, and we published many articles about this disease, but also we published about different types of parasites and some other pathologies present in, and also we performed some genetic analysis in the evolution of this marine bacteria, this marine brucella, compared with the terrestrial counterparts. This long-term effort exemplifies how a country with limited resources directly designed to study cetacean stranded has given light to disease conditions associated with individuals' mortalities, such as marine brucellosis in cetacean population in the Eastern tropical Pacific. Also, we have participated in the elaboration of books and a special edition of this disease for the World Organization for Animal Health, OIE. Robust diffusion of information through national newspaper and scientific publications have sensitized the population of the country on the importance of notification of the cases. We have learned that education is really important for the general public, and educate through good journalists and newspaper is critical. In our case, we found a really good journalist who respects the scientific part, and we call her when we want to publish something like this picture here. That was the first CT scan that we performed in cetaceans. And this was the time that CT scan was available for veterinary cases in our country. Now, we routinely perform the CT scan analysis to describe the pathologies in the brain and other organs. And how we work? Well, we take the CT scan results that are about 6,000 pictures and slides of the animal, send it during our night to Hong Kong to interpret the results. We got the results in our next morning prior to necropsy day. So we know, for example, the animal has broken ribs before opening it. And this is really helpful during necropsy because you can lose information, especially tiny fractures or bone tissue issues. And here is a model that currently we're working on, and this is the accumulation of cerebrospinal fluid in orange, which contains the bacteria that we're looking for. So we know in this case, for example, prior to necropsy, there is a lot of our sample, and that animal is extremely affected in the brain. So I have a question for you. Is there marine brucellosis in the Caribbean? The answer is we don't know. But we published this paper in 2009, over 10 years ago. And we described there that at least 33% of the stranded animal in the west coast of Florida, the Gulf of Mexico, has antibodies of Psela. So you may have this. This is a good example where storage from 1993 samples at Moe Marine Laboratory was a success samples to run after several years this kind of analysis. This presentation can be a baseline for the Caribbean and other small coastal communities through the globe to pool resources and joint scientific efforts for the conservation of these marine mammals. Of course, today I'm here, but there is a lot of collaboration, as I tell you before, to make this successful information that we got from our country. Thank you.